Oh, good. Good morning, everyone. Hope you all had a good weekend. The theme of today's lecture is, part of it is the intersection and interrelationship of politics and science. You will see this, if you guys have been following what's been going on in Arizona, we'll be discussing some of that just very briefly, and also we're going to get into stem cells today, which is talk about the epicenter of politics and science. What the advantage of today is you'll be getting real data and real logic and real information. And you have to sort that out from what you see out there. And we'll get into a little bit of that, just briefly. Anyway, so today we'll finish with human fertilization and blastocyst formation. Reader page is 47, but also this touches on development. So this kind of intersects a little bit with the next section on development. So pages 61 and 62 certainly relate to what we're going to discuss today and did on Friday. We're going to briefly talk about twins or twinning, then pregnancy, labor hormones, stem cells, and we'll probably get into development and cleavage reactions. Okay? So first is just to go over the menstrual cycle and the difference between high and low estrogen and et cetera, et cetera. It's very important to see this to understand human reproduction, in this case, the female reproductive cycle. In the menstrual cycle, in the beginning of the follicular phase, what you're doing is you're developing the follicles. And what's going on is you produce GnRH, gonadotropin-releasing hormone from the hypothalamus. The anterior pituitary produces LH and FSH, causes the theca cells that convert cholesterol into testosterone, and then testosterone into estrogen. At this lower level, so it's not nothing. It's just lower. Biologically, there's a difference between lower and higher for some systems. In this case, it's a very clear example. At this lower level, you have a classic negative feedback system. The object of what's the object of negative feedback? Keep things constant as much as you can, right? So at this lower level, if you have too much estrogen, you shut down LH, you try to shut down LH and FSH. If that gets too low, you bring it back up. And so you went within a window of acceptability. At this lower level of estrogen, this is precisely what's going on. Almost identical in feedback to what goes on continuously in the male system. Okay? However, an individual will cross a threshold, which is different for every individual. It's because one of the responses to estrogen is the proliferation of the granulosa cells, proliferation of theca cells. So basically, you have more cells and more estrogen. So the estrogen levels, although you're trying to keep it constant, within this window of acceptability, it builds up and builds up and builds up. You cross a threshold, and you convert from negative feedback to positive feedback. So at these higher levels of estrogen, higher, okay, versus lower, you then, instead of inhibiting LH-FSH, gonadotropin-releasing hormone, you stimulate the production. Because positive feedback, you amplify it. And as a result of this positive feedback, you get a surge in LH and FSH just prior to ovulation, and those two hormones, along with a little bit of estrogen, kicks on the ovulation process. Questions on lower versus higher estrogen? And again, there's a threshold that every individual crosses. It's a little different. I'm sure somebody knows the numbers on the average with 1 million women, for example, what that level is. That's irrelevant, what that number is. Those of you going into medicine might want to know that, but for the moment, you don't have to know it. Okay, questions on that? So you basically, females switch to a negative to a positive cooperative feedback. Then you have ovulation, which is what you want. The theca cells and the granulosa cells that are left form the corpus luteum, which is a differentiated tissue. This produces estrogen plus progesterone. The presence of these two steroids completely inhibits GnRH, LH, and FSH production. So it's not a feedback system, it's an inhibition. And that's what goes on in the luteal phase of the menstrual cycle, which is where the corpus luteum is around. Okay? So the presence of that other steroid just completely knocks out the production of GnRH, LH, and FSH. As you might predict, the basis of female contraceptives is estrogen plus progesterone. Individuals are different, therefore there's lots of different kinds of contraceptives out there with different concentrations. Okay? Questions on that? As you might predict also from this system, the basis of fertility drugs is enhancing GnRH, FSH, and LH, either production or activity, or you're actually giving women a form of some of those hormones. Okay? So make sure you know the difference between what would induce fertility versus contraception. All right? Any questions on that? Yes? Pardon?
So what induces fertility is estrogen?
No, estrogen plus progesterone does not induce fertility. Estrogen plus progesterone is a contraceptive. That's the opposite of fertility. Okay, what induces fertility is GnRH, LH, and FSH. Those are the basis of fertility drugs. Everybody clear on that? Okay, it's just the nature of the system. One is hormonally taking advantage of it from a pharmacological standpoint. Okay, make sure you know all those differences. Now, then we start talking about production of the follicle development that happens in the ovary. Females are born with primary oocytes. It gets ovulated. The process of the ovulation, you get that first meiotic division. You get a secondary oocyte. The secondary oocyte goes into the fallopian tubes where there's fertilization. And fertilization is defined as the ovum nuclei, which under the secondary oocyte undergoes the second meiotic division. You get a polar body in addition to this. You get an ovum nucleus and a sperm nucleus in the same cell. The nuclear membranes break down. The DNA is there, recombines, the nuclear membrane forms, and you get your diploid zygote. Part of this process, besides changes in respiration and massive changes in the cell, you have what's called a cortical reaction, and that is you have these cortical granules underneath the zona pellucida, which is the membrane surrounding the oocyte. These then merge with the membrane, changes the zona pellucida, so you no longer have the ability to have additional spermatozoa fertilizing this already fertilized entity. Okay? Does everybody see that? There's a change in the membrane. This zygote then undergoes cleavage reactions. This is a little bit of a development we're touching on here a little bit. And I'm going to leave this up throughout the lecture because this is very important for understanding twins and understanding stem cells. Part of it. You get a series of cleavage reactions, which you have partitioning of the cytoplasm, but you have mitosis of the nucleus. And you end up with an 8-cell morula. The 8 cells at this stage are completely indistinguishable, as are the 4 and the 2 and the zygote. Then you get a compaction around day 4, and about 3 or 4 days later, you get a blastocyst, which is an inner cell mass, and the inner cell mass will be very important in understanding stem cells, and a blastocele, which is fluid. And this blastocyst implants. One way to remember this is we'll talk about a blastula, and I think maybe in your lab you get a blastula. A blastocyst, a cyst forms an attached lump of tissue. So a blastocyst, it attaches. That's the way to remember this thing. There's an implantation process. The inner cell mass is about day 8 or so. Forms an epiblast, which forms the embryo eventually, and a hypoblast, which from an evolutionary standpoint, this is where the yolk would be, but mammals don't have yolk. But in fact, the original red blood cells that are produced come out of the hypoblast. This implants in the endometrium lining. Going back to the menstrual cycle, the optimal thickness for attachment to the endometrial lining is about 7 to 8 days after ovulation. And that's because it takes about 7 to 8 days to form this structure. So the whole system is timed. It attaches. It attaches at the trophoblast. The trophoblast is what's called extra embryonic tissue. It does not form an embryo. And this will be an important point in thinking about differences in stem cells. When this attaches, both maternal and fetal tissue comes together, you form a placenta. The placenta is a majorly important kind of organ during pregnancy. And it starts producing HCG, which we'll talk a little bit about in a few minutes. Human chorionic gonadotropin. It's a hormone that's uniquely produced by the placenta. and is also the basis of pregnancy tests. When that little line turns blue or pink, whatever it is, you're lighting up HCG. Okay? Questions on that? Yes? Say it louder, please.
So just a question about the zygote. When it goes from the secondary oocyte to the zygote, does it actually move like that?
No, does it shrink? No, I just drew it because I can put a 2 in that circle easier than I can over here. Okay, it actually stays about the same size. The question is, what's the size of the oocyte versus the zygote? Here I have it bigger, by the way. Okay, it's just the way I draw it on the board. Okay, don't get too worried about chalk drawings. I'm trying to do it so you can explain what's going on. Yes?
Trophoblast, the area between the placenta and the epiblast, hypoblast.
Yeah, the trophoblast is this whole area. There's actually a certain layer of cells that form the trophoblast, and it's right at the implantation point. Okay? Now, in terms of the politics, in the state of Arizona, they have decided, they have this abortion issue, and it doesn't matter what side of the issue you are on this. It's a religious issue, obviously. In the state of Arizona, they say 14 days before fertilization is where life begins. What is 14 days before fertilization? The primary oocyte. Scientifically, completely inaccurate and wrong. Okay? What is the chromosome DNA content of a primary oocyte, which is what all females are born with? 23, no. Primary oocyte. 46 chromosomes and 4 cDNA. Does an entity with 46 chromosomes and 4 cDNA make a human? No way. It then, after ovulation, you get a secondary oocyte. What is the chromosome DNA content of that? 23 chromosomes, 2 cDNA. Does that make a human being? No way. Proves Bio-1A class is smarter than the politicians of Arizona. All right? This is what the flow is in that entire logic. And I bring this up because you guys are all going to be voting. You guys are not quite 21 yet, right? Mostly. You guys will be voting in a year or two. Learn what goes on, what these guys talk about. You guys 21? What? Oh, you vote 18, sorry. When you vote in the next election. Sorry about that. It just ages me. Back in the days, drinking age was 18, voting was 21, so it's completely flipped around. Actually, for the good. All right. We'll talk about stem cells and get into this. So that's what's the flaw in this logic. When people look at this and just take non-scientific, illogical, wrong points of view on reproductive cycles. Okay, yes?
And it doesn't make any sense because if life starts 14 days before fertilization, then we're killing a baby every month.
Exactly. Not exactly. The question was, every time you ovulate a secondary oocyte. Right? And the other side of the coin is, what about the male primary spermatocytes? How many are we killing? So the point is, the timing of this law, by the way, was really kind of interesting relative to where we are in this class. You completely understand the flaw in what they're talking about. There's political reasons why they did it. I don't want to get into it right here. But the scientific reasons do make zero sense. Okay, anyway, so you get the cleavage reactions, and we will talk about that. All right? Any questions? Okay, so let's talk about twins briefly. I have a set of twins. I was going to bring them here today, but they have school today, so they'd be running around too much. Set of 10-year-old twins. Different kinds of twins. Okay, what I have is fraternal twins. And fraternal twins involve 2 ovulations, involving then 2 fertilizations, okay, and 2 sets of development. Fraternal twins are like siblings, of different ages. They come from 2 separate zygotes, or I should say 2 fertilizations, 2 zygotes. And these are reasonably common. Women, if you correct for in vitro fertilization, the optimal time that women produce more than one ovulated secondary oocyte is late 30s to late 40s in that time frame. And so any of you that have either siblings or parents that you know, or uncles and aunts that have fraternal twins, you should find out if they didn't do anything in vitro. You have to know them well enough to ask them that question, I guess. How old were they? And it tends to be a higher risk of fraternal twins, or triplets even, late 30s to late 40s, which is about 5 to 10 years before menopause. So basically, you got two of these going on. Fertility drugs, when fertility drugs amplify this system, and they have multiple ovulations, what you tend to have a lot of times is triplets, quadruplets, which are all fraternal, because it's due to multiple ovulations. And these are like siblings. Everybody see that? In contrast, identical twins are cells in the cleavage stage. Now, in the cleavage stage, from 1 to 8 cells, they're completely indistinguishable. And they're each capable of forming another baby. The most common type of identical twins is you have the zygote, and you make the two cells, and they don't come together very well for whatever reason, or they come together and they fall apart. Then these then go on to the cleavage reactions to forming the marullas. And you can take, and this can happen at the 4-cell stage, one could break off. It could happen at the 8-cell stage. As long as it's going on before compaction, which is when all these guys come together very tightly, you can get identical twins. How do you think you can get identical triplets? Which also can happen. Pardon? Keep going. Doesn't break 3 times. Think of your math. You know, you never get mitosis that goes into 3 cells. Yes? That's one way. That's very good. You could say you could do it twice and one dies, but what if one doesn't die? Keep going. You're on the right track. Yeah? Yeah, so you can have identical triplets. It's like you're both ways you said it. Break twice, one dies. That's fine. That works. The more common way is one of these guys does this again. And here you can get, instead of this thing here, you get triplets, identical triplets. How can you get triplets that are one set identical and one set different? How's this for math? So you have triplets, but 2 are absolutely identical to each other, and 1 isn't. What do you think happens there? Yes?
Fraternal twins, and then one of them splits.
Right. You have 2 fraternal twins, one of them splits into identical twins. This can definitely happen. I've seen it. Not personally, but I know someone that has triplets, 2 of which are identical. Okay? So keep that in mind. Okay, now you can also have what's called mirror image twins. Mirror image identical twins. And what is here is it's genetically identical. They're genetically identical, but they're mirror image. So, for example, one can be left-handed, one can be right-handed. The most famous example of that are the Bryan twins that are doubles, U.S. doubles champions. They're obviously doubles, they play tennis. The ideal doubles combination is where one partner's left-handed, one partner's right-handed. These guys are genetically identical twins, except one is left-handed, one is right-handed. Okay? You get mirror image identical twins when it splits between the compaction stage down to the blastocyst stage. At early blastocyst, it can start splitting. They're genetically identical, but they tend to be mirror image for reasons nobody truly understands. Okay? You can also have problems because depending on where it splits, you can have 2 different placenta, which is the best thing, but you can also share placenta, or you can even share embryonic sacs, depending on where it splits. In the case of co-joined twins, which are Siamese twins, that's called a sort of common name out there, those are just mirror image twins that never completely split apart. So those are all mirror image twins. So it's because it happens later in this process. So there's just enough differentiation going on that one can distinguish the two, being left versus right-handed, but they're genetically distinct. I mean, excuse me, they're genetically identical, but they're distinct in some other parts of their phenotype. Okay? Questions on that? Does everybody see that? The co-joined twins or mirror image twins, they just never completely split apart. It's not all that common, but it's out there. And those of you going into medicine will, pediatrician type things or obstetrician, things of that sort, will see this out there for sure. Okay? Any questions on twinning? And I'll keep this on the board here, because when we talk about stem cells, for all the reasons you can get identical twins that will work and so forth, it's the same thing, underlying principle with the stem cells. So I'm going to leave this up there. Okay, so after the blastocyst comes in, attaches, you get the trophoblast, you get the placenta formation, you make HCG. It's not just a random hormone. Humans did not evolve, hundreds of thousands, whatever, years ago to make HCG to then use as a pregnancy test starting in the 1950s or something, or the 1960s, right? There's a reason for HCG. And that is, in the first trimester, pregnancy is put into 3 trimesters. In the first trimester, you still have the corpus luteum around for the first 3 months. And the placenta makes HCG in order to maintain the viability of the corpus luteum, to keep it around. Without pregnancy, without HCG, the corpus luteum disintegrates or degrades. And that's actually what happens at the end of a menstrual cycle when you have no pregnancy. There's no HCG, and therefore the corpus luteum falls apart, and then the whole cycle happens again. So it has a positive effect in viability. Now the corpus luteum, what hormones is the corpus luteum producing? Estrogen and progesterone. Well, the reasons for estrogen and progesterone is in part to prevent the anterior pituitary. Let me just put you inside the anterior pituitary here. It is in part to prevent LH, binds to receptors, of course, prevents the production of FSH and LH. That maintains itself. But in addition, there's a positive reason for having estrogen plus progesterone, and that is when it goes into the cell, it causes the anterior pituitary to make more of a hormone called prolactin, which is important in another organ that starts developing during this time. It binds to its receptor on the mammary gland. The mammary gland during pregnancy has to start proliferating, developing, and eventually you want to make milk proteins. Prolactin is one of the hormones that stimulates the production of the milk genes. Casein. You don't need to know beta casein, but it is the milk protein. Progesterone also plays a role in that. So the estrogen plus progesterone also has a positive effect on the mammary gland. Okay? So it's estrogen, progesterone, prolactin, amongst other hormones, but these are the main ones. Every time I have an arrow here going into an organ, that organ responds because it has what? Receptors for the hormone. There we go. Now the placenta also makes another hormone called CS. And let me just write this out for you. CS is chorionic somatomamotropin. Just from the name, somatomamotropin, growth of mammary cells. The somatic cells in the mammary, so your typical cells in there. This is very important for keeping the mammary gland going. So already you can tell from this that the placenta is an endocrine gland, amongst other things that it does, because it's making, just on this first trimester, makes HCG and CS. Questions? Yes?
So in addition
Correct. The question is, does estrogen and progesterone directly target the mammary gland? Absolutely. Absolutely, yes.
Why did the mammary gland development so early?
Why does it start so early? It's the way we've all evolved. I can't give you a real answer to that. It takes a long time for it to develop and grow, and get the cells ready to differentiate to produce milk. I think what it is the whole system is kind of lined up to get going. Because what if pregnancy is shortened by a month or two? It can happen, right? The mammary gland can still produce milk then. I mean, that's one way of thinking about it. But why human gestation is 9 months versus, I don't know, mice or what, a month or something like that? 21 days, close. I mean, why is that? Why do we need so long to do it? I don't know. I honestly don't know. It's the way evolution took place. But the point is, the reason the mammary gland develops, the reason is because of the hormones that are produced. Okay? So it's not just fortuitous that the mammary gland starts developing at that point in time. It's because of the hormones that are produced, in this case, by the corpus luteum and the placenta. Now, what distinguishes the first trimester from the second and third trimesters, I'll do this up here. Wait a minute. I'll do it here. Second and third trimesters. And there's all sorts of differences in growth and development of the fetus, blah, blah, blah. We don't need to worry about that too much. But in terms of the hormonal mechanisms, the corpus luteum does in fact break down. And what kicks up the difference in all these hormones is the placenta. And that is because the placenta, it breaks down because the placenta stops making HCG after about 3 months. By the way, it is HCG that causes the morning sickness, or one of the main triggers of morning sickness. You can talk to your moms if they had morning sickness, if it stopped about 3 months. That's why it's such a relieving point of view. And if you have twins, you have even that more. If you have 2 placentas with the twins, which is usually the case, you have even more HCG and even more difficult morning sickness. The placenta, however, you need estrogen plus progesterone. And estrogen plus progesterone is then made by the placenta. So the placenta picks up the difference. Now, the key thing here, is that the placenta is a huge, huge organ compared to a lot of these other corpus luteum. So if you look at the trimester, here's time, magnitude of the hormones. This is coming from the placenta. First trimester, HCG comes up and comes down. And then estrogen plus progesterone kicks up. The most progesterone and estrogen a female will ever make is during the second and third trimesters, due to the placenta, which is huge. It's almost as big as the baby when it comes out. Okay? In fact, it surprised me on my own kids. I was there. I cut the umbilical cord, actually, for all 4 kids. And it surprised me this first time that the placenta was so big. So it's a huge organ. The advantage was there's a lot of tissue there. A lot of the discovery, a lot of these hormones came from isolated placenta because there's so many of them and so much tissue. So it was actually experimentally a very important kind of tissue. All right? Questions on this? All right? Then, after all this, we have labor. And I'll bring that thing down in a second. You also have hormones during labor. So what happens is, from the ovary, does have some estrogen gets out, of course, and it goes to the uterus, has its hormones, receptors rather, and it stimulates the production of a receptor, receptor production of a hormone called oxytocin, which comes from the posterior pituitary. Okay? So what happens is this oxytocin will hit the uterus. In addition, this oxytocin also hits the placenta. And it causes the placenta to produce another receptor, another hormone called prostaglandins, which is a lipid-like. When oxytocin is produced, the combination of oxytocin directly on the uterus and oxytocin going to the placenta to cause the production of prostaglandins causes contractions. And the contractions, which create pressure, then go up to the posterior pituitary, and a positive feedback causes the posterior pituitary to produce more oxytocin, and therefore more prostaglandins, and therefore more contractions. Therefore, more pressure, more oxytocin, more prostaglandins, and keeps going until the delivery. Okay, so as you can imagine, if you're going to develop a labor-inducing hormone, what would you be giving someone who's having trouble starting up labor? Yeah, some form of oxytocin and prostaglandins. Usually it's a synthetic form. Okay? If you wanted to stop labor, what system would you hit here? You might try to shut down the production of oxytocin or prevent oxytocin from working. Okay? So again, just those of you get into this a little more directly and career-wise, all you're doing is going on either side of this hormonal balance at this point in time to either induce labor or stop labor. You might want to do that at different points in time, depending on what's going on biologically. Okay? Questions? Now, this whole thing relates to stem cells. This whole system relates to stem cells. I'll do this here. And have you guys heard of stem cells, right? Loosely, I don't know if you had it here in this course. Certainly you get it in the news quite a bit. It's a political hotbed in a very interesting way. Stem cells are basically undifferentiated cells that then differentiate into different cell types and tissues, but they also can self-renew. Self-renewal. This is all happening through mitosis. And you have a store of stem cells as adults that are very important in the way you function. Just getting some more chalk here. Okay? The political issue / scientific issue is it possible to take a stem cell and in the lab give it a certain differentiation so that you get a specific tissue type that you need? Such as one example where they're trying very hard is people that are, say, paralyzed due to a spinal cord injury or a nerve cell injury. Can you then, in theory, make a stem cell, patch it in, and get them to walk again, for example? And that's why there's so much attention being paid to this. Now, where it gets muddled, certainly in the press, in the political side as well, not so much the scientific side, is that there are different kinds of stem cells. And depending on what kind it is, changes what you can do with them. So one of the types of stem cells is called totipotent. Totipotent stem cells can go to all cell types, both embryonic and extra embryonic. In humans, the only true totipotent stem cells, the only ones, is from the zygote to the 8-cell morula, which you should immediately recognize as the cells that can do what when they split apart normally? They make identical twins. Okay? So in humans, the only ones is the zygote to the 8-cell morula. All these cells are indistinguishable And they all can eventually form blastocysts and give you, like, for example, trophoblasts versus non-trophoblasts. Okay? Sometimes you get that muddled out there. Another kind of stem cell is pluripotent, which is where most of the activity is. These guys can only make embryonic tissue. So in this case, a pluripotent stem cell could not make a trophoblast. You can't make a complete and other kinds of extra embryonic tissue. What are the sources? Well, one source is the inner cell mass of the blastocyst. So one source, one very important source, is this inner cell mass here of the blastocyst. And these are called out there ES cells, for embryonic stem cells. Okay? There's two other sources that you don't see done very much, but they're certainly there. You also have embryonic germ cells. And these are the precursors for the spermatogonia or the precursors for the oogonia. These are all diploid cells. Those are obviously hard to get at. And ones you probably would never really want to use for stem cell work, but some of it you have. Embryonic carcinoma cells. And a typical source of this would be gonad tumors of the fetus. All of these are cells of importance. The reason is, but it's the ES cells from inner cell mass are the most important because they're normal, somewhat normal-like. A pluripotent stem cell could go to any cell of the embryo. And therefore, it's a good starting point because, in theory, whatever you need, you should be able to give it the right kinds of signals, in theory, and get the tissue that you want. Easier said than done. They're just at the tip of the iceberg. But that's where they get the inner cell mass. As we'll talk about in a few minutes, with the inner cell mass, you can do this completely in vitro. And that's part of the scientific interest and the political controversy. Okay? Let me do this over here. You've got two other kinds of stem cells that are very prominent in adults. Now, adults, all of you sitting there all have various kinds of stem cells. These are called multipotent and unipotent. And I consider these really one and the same category. Multipotent and unipotent have a restricted cell types that they can differentiate into. In the case of unipotent cell types, you go into one. So an example here would be muscle stem cells. Only can go into muscle cells of the adult. Multipotent can go into multiple cell types. So the only difference between multipotent and unipotent, they're both adult, is one goes into one, the other can go into more than one. So in the case of and I'll put this down in a second here. So in the case of multipotent, a good example are bone marrow cells, which we'll talk about next week. Bone marrow stem cells can go into myeloid branch of stem cells or lymphoid. And you'll see this in more detail next week. As you can see, two distinct cell types. The problem with the nomenclature, unfortunately, the original name for these bone marrow stem cells is pluripotent stem cells. And you just got to almost ignore that. What these are an adult stem cells. They are clearly only multipotent because they have a restricted cell type by which they can differentiate into. Pluripotent stem cells can go to any, in theory, any cell of the tissue. So when you read it in the books, it's even in the books, sometimes the bone marrow stem cells have this incorrect name. Okay? Questions on that?
What do those two words say?
Which two words? Oh, okay. You'll get this next week. myeloid and lymphoid. Okay? We'll worry about that next week. Myeloid cells are formed like red blood cells. Eosinophils, basophils, lymphoid stem cells form lymphocytes. But we'll talk about this next week. Okay? But these are examples. You have many examples of these kinds of stem cells. Now the issue is the following. Here's the political issue. Just put this up here. You can in vitro, completely in vitro, you can make a zygote, fertilize in vitro, form a morula, and get a blastocyst. You can then take out of the blastocyst the inner cell mast cells. This can be done, and you can put them in cell culture. Okay? And in theory, and this is what people are trying to do, is, remember I say you are your hormones, right? Well, you give them hormones, certain kinds of genes, various signals, signal transduction, et cetera, and in theory you can change this to a specific differentiated cell type. Okay? And then in theory, put this back into the patient. And the advantage of doing this, the scientific advantage, is you can get a lot of inner cell mast cells. And what's nice is they're pluripotent. Because if you take something like a muscle stem cell, it's very hard, it's been impossible to this point, to turn it into something else. But people have had limited success in some areas of turning these guys into nerve-like cells and other kinds of cells that are out there. So we're learning a set of rules how to do this. Okay? Now, you can take stem cells from an adult. The advantage of an adult is, in theory, you can take your own stem cells, in theory, change them into something else and put them back in your own body. In theory. The advantage being, you're already your own cells, so you don't have any immune response to your own cells. We're not that far yet. There are some examples of that. The downside is, in the adult, you don't have all that many stem cells. You can get a lot more of these in vitro fertilized ES cells than you can ever get in an adult. Now, the political controversy of this is you have these in vitro fertilized cells. Say you go to the 8-cell morula. This is all done in vitro. You get a blastocyst. And you have two choices here. You can continue on and make the fetus, or you can make ES cells and use it for this kind of analysis. So how does one get around this controversy? Because some people don't agree of using it this way. Well, you can get around it by when you make the morula. So here the 8-cell morula. There's various ways of doing this. Some of this has not worked in humans. Some have worked really nicely in mice and primates. You have this 8-cell morula. You can take out one cell. Remember, up to the 8-cell morula, all these cells are completely indistinguishable because you can still make identical twins, even at that stage. So one thought is you can take one cell and make your embryonic stem cells and do your experiments, and take the rest of the morula and make the fetus. So you haven't gotten rid of a fetus. You've simply taken a cell and you've done something with it. And one of the futures might be, this is all science fiction at the moment, but not too far off, you have a baby, or you have done it in vitro. You take one of the cells and you store these stem cells, because just in case your kid has a serious physical or physiological problem later on that stem cells would help, you'd have frozen down those stem cells for them to use. So you can easily make the argument that when you have a pregnancy, maybe you get a hold of a few stem cells, store them for later use. Now, another source of some of these stem cells, they're not quite pluripotent, but they're close, is the amniotic fluid. Okay, the fetus is sitting there in the embryonic sac. There are stem cells there. And in theory, you could do the same kind of thing, is grow ES-like cells from that. Science fiction at the moment, but we're really not that far off. Okay? A lot of these are issues you're going to be confronted with in your careers. Now, there's other approaches. There's other approaches here. Another approach is called, that has worked in primates. Primates only, and there's also a problem. And that is it's called somatic cell nuclear transfer. Okay? In primates, you can take a primary oocyte and take out the nucleus. I'll just give it a nucleus here. It's a primary oocyte. Enucleate it. Okay? Then you give it a diploid nucleus. In the case of primates, I think they used a skin nucleus of some sort. So you give it a nucleus that's diploid, and you can get a zygote. Okay, so this actually works in primates. The problem is, and they haven't been able to correct this at all, is you get extra chromosomes made. You get aneuploidy. You get extra chromosomes, or sets of chromosomes. And in cancer, one of the big issues is aneuploidy. You get extra chromosomes in these cells. So this has not worked, but in theory, it has the chance of working. Another way to do this, you can reprogram cells. You can take in mice a skin cell, give it a couple genes. I think there are 4 different transcription factors you can give it, and you can get an ES-like cell, so something that's close to a pluripotent stem cell. So the other area people are working on is to reprogram normal cells to become stem cells. And this is a big issue also in cancer, because if you have breast cancer and it's estrogen receptor positive, what do you treat that we've talked about? An estrogen receptor positive breast cancer cell. What can you treat it with to stop the growth of a cell? An anti-what? If it's an estrogen receptor positive cell. You can treat it with an anti-estrogen, right? Like tamoxifen. Well, eventually in the tumors, eventually the cells grow out that are no longer estrogen receptor positive. And it's thought that's because of the stem cells sitting there growing slowly, not responding to the therapy. And then you get rid of all the estrogen receptor positive cells through tamoxifen and similar kinds of treatments. And what's left is a stem cell or two that's estrogen receptor negative. It grows out, becomes a tumor, and it's no longer responsive to estrogen antagonists. It is now independent of that and a much more advanced kind of breast cancer. And it's because it's thought because of the stem cells are there and they grow out of the population. Okay? So it's an issue also in terms of cancer therapies. So you'll see this quite a bit professionally. Questions on stem cells? Questions? Okay, we don't have enough time to start in development, which we'll do Wednesday. But the point is, we've actually done a part of human development. And the one thing to take home is during the cleavage reactions, just remember, in humans, the cells are indistinguishable up to the 8th cell morula. And if you know that, you know a lot about human development. Okay, so we'll see you Wednesday.